Hi, my name is Austeja Ciulkinyte. I am a PhD student at the University of Edinburgh. This here is my poster representing a small side study that I've done as part of my PhD project. This project stems from the idea that we use mouse models a lot in neurodegeneration research, and we know in particular that blood vessel dysfunction is a very important contributor to neurodegeneration. However, we haven't really fully understood how well does mouse aging reflect human aging and whether the processes that underpin aging in mice are similar to the ones in human. And so this is what this project is all about. So I look at a cohort of young and aged mice as well as young and aged humans. I investigate the proteome of the whole brain as well as the blood vessels isolated from the brain. This is an example of what those blood vessels look like. And I ask how well does our mouse aging match human aging? What we see is that if we look at whole brain aging, we see a fairly good overlap. So that means that mouse get older in a similar molecular pattern as humans do. However, if we look at the blood vasculature in isolation, we now start to detect proteins that are actually regulated in completely opposite directions in humans and in mice with advancing age. These proteins often have to do with the cytoskeletal organization of the cell as well as the protein production and turnover of the cell, which can mean that these might be really interesting pathways to investigate if you want to understand why humans get some diseases such as stroke and Alzheimer's disease, whereas mouse don't.